This is the first lecture in Unit 1 in the microbiology course. There are several links that are presented in this lecture that have videos. The videos are going to be available under a section under Unit 1 that will say videos and websites. There are links where all of the videos are embedded, as well as you'll be able to link to them off of your notes if you download these as the Word document. The history of microbiology begins in the early 1600s. Microbiology is the study of microbes or microorganisms, which include all organisms too small to be seen without a microscope. The microbial world was discovered by a Dutch lens maker, Antony van Leeuwenhoek. He lived from 1623 to 1723 and called these microbes beasties. The protozoa were discovered in 1674 and the bacteria in 1676. His early microscope was really nothing more than what we would call a magnifying glass today. You should also be able to see these links and be able to link to them directly off of this Tegrity recording. Microorganisms fall into a few different categories. The taxonomic system used was developed by Carolus Linnaeus, who lived between 1707 to 1778. This system was developed for naming plants and animals and grouping them together. Prior to this, naming and classification varied greatly from country to country, even scientist to scientist. A modified Linnaean system is still used today, although there is some reclassification occurring with DNA technology, showing us new relationships between organisms. This is the basis for our major groupings, the eukaryotes, prokaryotes, and archaea. You will actually find a lot of reclassification occurring in microbiology. There is also a video here that you can link and watch on Carolus Linnaeus. The first group of microbes are the fungi. Fungi have cell walls and obtain their food from other organisms. They are eukaryotic cells because they contain a membrane-bound nucleus. The fungi include both the molds and yeasts. Molds are multicellular organisms which grow as filaments called hyphae. These are commonly seen growing on foods like bread and cheese, and the hyphae give them a fuzzy appearance. The yeasts are unicellular and tend to be oval or round in shape. These typically reproduce by a process called budding. Though the protozoa are single-celled or eukaryotes, they typically live in water. Some will live in the moist environments of an animal host. Algae are organisms which can be either unicellular or multicellular and are able to do photosynthesis like plant cells. Prokaryotes are unicellular organisms with two groups. The bacteria have cell walls made with a special material, peptidoglycan. The archaea are similar to the prokaryotes but do not have peptidoglycan and live in extreme environments. The question of where life originated from is not a new one. Some of the early ideas by scientists were things that we would consider absurd with our knowledge today. One of these such ideas was spontaneous generation or abiogenesis, that life arises from non-living things. Several scientists in different countries all made contributions in disproving spontaneous generation over several years. Francesco Redi, an Italian scientist who lived from 1629 to 1699, showed that decaying meat which was isolated from flies did not have maggots. John T. Needham, a British scientist living between 1713 and 1781, worked on this theory as well. He prepared boiled gravy infusions and sealed them. He observed them days later and noticed they were cloudy and attributed this cloudy growth to a spontaneous life force. Part of the problem with Needham's experiment was that he did not boil the gravy infusions long enough to actually kill all the organisms, so naturally they would be cloudy in a few days. Italian scientist Lazzaro Spallanzani, who lived from 1729 to 1799, also boiled gravy infusions, but in his case he boiled them almost an hour. This time the closed vials stayed clear, and those exposed to air became cloudy. This led to an idea that there was something in the air causing life. Louis Pasteur, who lived from 1822 to 1895, performed an experiment similar to Spallanzani, but used a curved-neck glass. This allowed the infusions to still have air or be aerobic, and all his nutrients were available as well. But this prevented airborne microbes from landing in the infusions. His result disproved the idea of spontaneous generation. This was not his only contribution. He also made the discovery that anaerobic organisms could ferment grape juice, and that you could pasteurize the grape juice by heating it just enough to kill the bacteria. These discoveries are still used today in the wine and food industry. There are several links here that lead to a Discover video that's on YouTube that is actually cartoon, family-friendly video about Louis Pasteur. I would strongly recommend watching these videos because it does make it a little bit more interesting to learn about these historical figures. The scientific method is the technique used for making observations and finding answers to scientific questions. The purpose of using this technique is to minimize errors and misinterpretations of results. Initially, the idea is tested in the hypothesis. Control groups with a known outcome are used as comparisons to the experimental groups who receive the experimental treatment and have an unknown outcome. After verification by many scientists and experiments over the years, the hypothesis becomes accepted as theory or law. Even with the scientific method, science can only rule out possibilities, but can never determine something to be true under absolutely all circumstances. Again, there is a video link here that is much shorter than the last one. The study of biochemistry or metabolism also played an important role in the current understanding of the microbial world. The German scientist Edward Buchner, who lived from 1860 to 1970, demonstrated the presence of enzymes and showed fermentation did not need living cells. This was important in beginning the field of biochemistry. Another one of Pasteur's contributions to science was the germ theory of disease in 1857. This hypothesis states that microorganisms are responsible for disease. The pathogen is the specific organism that causes disease. This was the start of the field of etiology, which is the study of causation of disease. Robert Koch, the German scientist who lived from 1843 to 1910, investigated etiology. He studied infected animal blood and demonstrated when the animal was given a pathogen, it would cause specific diseases. He developed Koch's postulates as steps used to prove the cause of any infectious disease. The postulates are as followed. The suspected agent must be present in every case of the disease and not in healthy hosts. Second, the infectious agent must be isolated and grown outside the host. Third, when the infectious agent is given to a healthy host, they must develop the disease. And fourth, the same agent present in the original diseased host must be present in the experimental host. There are some limitations to using these postulates in real-life situations. There are some who feel the germ theory of disease is false. Keep in mind the germ theory is meant to be applied to diseases of infectious nature, not degenerative, traumatic, or genetic nature. Also, there are ethical concerns involved in using Koch's postulates. Harming healthy humans is not considered an acceptable practice, and some will even argue against using animals for this purpose. Several other scientists contributed to our knowledge of the microbial world. Charles Laveran, who lived from 1845 to 1922, demonstrated the protozoan cause of malaria. Edwin Klebs, who lived from 1834 to 1913, described the bacteria causing diphtheria. Dmitry Ivanowski, who lived from 1864 to 1920, and Marthas Bejerink from 1851 to 1951 demonstrated a viral disease in tobacco plants that could pass through a filter. Walter Reed, who lived from 1851 to 1902, proved in 1900 viruses cause disease. Danish scientist Christian Gram, who lived from 1853 to 1938, developed a stain using a series of dyes to separate bacteria into two groups. The Gram stain is commonly used today. The Hungarian Ignaz Semmelweis, who lived from 1818 to 1865, was the first to require hand washing with chlorinated lime water to eliminate cadaver particles, which were thought to carry disease. English scientist Joseph Lister from 1827 to 1912 was the founder of aseptic surgery and used phenol as an antiseptic. The English nurse Florence Nightingale, who lived from 1820 to 1910, was the first to introduce the use of cleanliness and aseptic technique into nursing. While some of these things seem quite obvious to us, early attitudes viewed the situation more along the lines of, why clean the instruments, only have to have them get dirty again. The use of aseptic technique in hand washing is one of, if not the greatest contribution to improving the survival of surgery and infectious diseases. Here is a link to a Florence Nightingale animated cartoon on YouTube. There are 5 parts to this link. I do not own these links, so there is a chance that they may not work after some time. So there is an alternate version on here as well that you can try. I believe they both have the same content. One just has much longer video clips than the other. The English scientist John Snow, who lived between 1813 and 1858, studied cholera in contaminated water. This provided the documentation needed for clean water and sewage treatment. Another English scientist, Edward Jenner, who lived from 1749 to 1823, studied immunology and vaccination. He used the pus from cowpox lesion to inoculate his son. His son developed cowpox but became immune to it in the future. Paul Ehrlich, who lived from 1854 to 1915, studied chemicals that killed microbes differently but not people. This started the early study of chemotherapy. While it is common for people to associate the term chemotherapy with cancer treatments, it actually refers to the use of chemicals as therapy, and so it includes a much broader range than just cancer or antibiotics. The Golden Age of Microbiology is considered the time period between 1857 to 1907, with the work of Pasteur all the way up to Fleming with pharmaceutical microbiology. The work from this time period gave us the study of immunology, or the specific defenses of the body against pathogens. It also has made us aware of nosocomial infections, which are those acquired in healthcare settings. I imagine there was a large amount of these back in the time before they felt the need to clean instruments in between patients. There are other scientists who made important contributions in this field that are not mentioned here, since this is just an overview of microbiology history. Our knowledge of microbiology continues to increase after the Golden Age. Albert Kluver, who lived from 1888 to 1956, and C.B. Van Niel, who lived between 1897 and 1985, proposed that bacterial biochemistry is shared by all living things. You will notice much of the biochemistry of bacteria is the same as what you've already studied in humans today. You will get to revisit glycolysis, the Krebs cycle, and the electron transport chain, as they will occur in many bacteria as well. The geneticists Oswald Avery, Colin MacLeod, and Macklin-McCarty determined genes contain molecules of DNA. George Beadle and Edward Tatum established genes actively relate to the function of protein encoded by that gene. This has led to the field of molecular biology, which combines the study of biochemistry, cellular biology, and genetics. In 1965, Linus Pauling proposed the idea that a gene sequence could provide understanding of evolutionary relationships. This study is leading to changes in previously described relationships between organisms. In the 1970s, the work of Carl Boyce made a significant influence in the determination that nucleic acids reveal three major groups of organisms: the bacteria, archaea, and eukaryotes. Just a short time ago, in 2003, we sequenced the entire human genome. The field of recombinant DNA technology involves the use of genetic engineering and gene manipulation. Gene therapy is a process of inserting missing genes or repairing a gene in human cells. Much of this work is tied in with the studies of stem cells. Environmental microbiology studies the role of microbes in our environment. Bioremediation uses living organisms to detoxify polluted environments. We are able to use this technology to process sewage enough to make new drinking water. There are even places in Tacoma that are able to process their sewage enough that you can drink their water.